What's up, everybody? We're back doing another review video. Today, we're going to be getting into cells and cell theory. This is page 6 of our review. We're just going to do the best we can. We'll talk through our stuff just like normal, and hopefully, this turns into something that's going to be helpful for the EOC. So, we start here with cell theory. It starts by asking us the three parts of the cell theory. It is fairly common that they end up asking you a cell theory question on the test. Most commonly, they'll give you a multiple choice question where they'll list the three parts of the cell theory and then add in a fourth point that's not part of it, and then have you identify what is not part of the cell theory. So let's take a look at what these are and see if we can get our explanations here. First, all living things are made of cells. Second, cells are the basic unit, structure and function in life. And third, cells come from pre-existing cells. Now, just on face value, these three statements aren't all that wild, but we kind of need to make a big point here because if they don't just ask you about the parts of the cell theory, they're going to be asking you why it's a theory and not a law. So let's get into the difference between a theory and a law real quick. If we set up a T-chart, I cannot draw with my mouse very well. Law and theory. So the big differences we're having here, both of these, because they are involved with science, have to do with evidence. But the amount of evidence or the way we use the evidence is what the actual difference is here. When we talk about a law, we are talking about something where we have all the evidence. We've tested, we've looked for, we've found everything that we needed to. The second thing about a law is that we can use that evidence to prove it. Now, the reason that that's significant is when we work in science, we need evidence for everything. We want to back things up. That's why we do experiments and test things. We need to be able to go, I don't just think this based off of this information and this evidence. This is true. The problem with the cell theory is we make some wide sweeping claims that even though we have some evidence, we're not able to actually prove. For example, all living things are made of cells. Okay, well, we definitely have evidence for that. Everything that we've looked at so far, everything that we keep finding, turns out they're all made of cells. That's great. But what about stuff that's gone extinct already? Did you check those guys? Oh, well, they're not really around anymore, so like it's kind of hard to check that. Okay, so then that means it's technically possible that they wouldn't be made of cells. Cells come from pre-existing cells. Okay, what about the original cell? How'd that get there? Well, we don't exactly know, but every cell that we're aware of now has come from a cell that was from before it. Right. So what you're saying is you can't actually prove it. A theory is something where best way to describe a theory, at least in my opinion, a theory is the best explanation based on the evidence we have. And with cells, we can do that. Everything that we have coming in, all the research we're doing would back up the cell theory, but we don't have all of the evidence about things from the past. And because of that, we can't go as far as calling it the cell law, because technically, this is just based off of the information and the explanations and evidence that we do currently have now. So between those parts of the cell theory or trying to figure out the difference between a theory and a law in reference to it, that's more than likely what would be coming up on the EOC if they ask you questions about cell theory. What discovery and further development led to this to these scientists finding evidence for the cell theory? That would be the microscope. Now, That as a question of like, what was the discovery? I don't believe that's going to make it to the EOC. But in reference to the types of microscopes, like we're going to be doing down here in questions 4 and 5, always seems to make it to the EOC. So we'll tighten those up in a moment. Question 3, explain how the cell theory has changed over the years. Well, first thing we have to say, the cell theory was not made up by one person. Cell theory was organized using info from many different scientists over time. Second, how has it changed over time? Well, like anything in science, if we come up with new evidence or new information, we're going to adjust what our explanation was to look at it in light of that new evidence. So, how has things changed over time as far as the cell theory goes? Each time we discover something else, each time we discover something or gain new info, we adjust our theories. Now, we haven't discovered anything that has contradicted the cell theory yet. We just keep coming up with more evidence for things. Before, when they were talking about cells, they didn't necessarily know of all the different types of cells. They had seen plant cells and bacteria cells and animal cells. But as we brought things in like fungi and protists and other things, over time, we've had to kind of adjust things a little bit. That's why it kind of seems like the parts of the cell theory might seem a little bit general. That's because of how many different pieces of information we got. We have to make sure that the statements we use can be proven and true. If we can't prove it all the way, then we make our best explanation based off of the evidence we have, and we would call that a theory. So definitely going to be a question from somewhere in this info that we just did on the EOC, but we'll see which part it is. Sliding down here to our microscope section. Which microscopes would you recommend to a researcher use to work with living things? Well, first, let's just recap our types of microscopes. I think I'm just going to set up a paragraph on the side over here. This is something that would be a good idea to put into some margin space or maybe write down in another place. But there's going to be 3 main types of microscopes that we work with. We have the compound light microscope. And these are the ones that we have at school. The big thing about the light microscope, we can get about 1,000 times magnification from a light microscope. That's great. That's actually zooms in quite a bit. We can get a whole bunch of work done with that. They're relatively inexpensive and they're readily available, which is great. There's a little bit of a downside though. The stuff that we look at has to be very thin. Specimens must be thin. We often shave things as thin as we can and put them on slides to look at under the microscope because we would need the light to be able to go through them to help us see the structures that are inside. So that's not so great. To answer this question, for question number 4, the answer is going to be light microscope. And this right here is the reason why. Since we're shining light with a light microscope, this means we can use it to watch living processes. I can watch a cell do mitosis under a light microscope. I can watch other forms of cell division under a light microscope. I can watch little single-celled guys that live in pond water go around and find their food and eat things under a light microscope. Light is not going to be damaging or harming any of these things, whereas our other types of microscopes aren't using light. Let's bring these guys up. We have electron microscopes, and there's 2 main types of these. We have a scanning electron microscope, and we have transmission electron microscope. Now, these two work in similar ways. Instead of shining light onto what it is that we're looking at, we blast electrons at them. The reason for this is electrons are very small and their wavelength is smaller than the wavelengths of light that we would be using, meaning that we can fit more of them into these super tiny areas and therefore zoom in farther and still be able to get a picture. So electron microscopes can actually zoom in at least 10 million times, which if we're comparing that to what we had before with 1,000 times the light microscope, that is significantly more magnification. Now, a scanning electron microscope and a transmission electron microscope are very similar. And the good news is their names kind of describe what they're for. A scanning electron microscope is going to bounce electrons. We use E minus for electrons as an abbreviation. Bounce electrons off the surface of the specimen. This is going to give us a picture of the outside. So if I was trying to see what the outside of a cell looks like, if I was trying to zoom in and look at the exterior structure of something and see what it's made of and what pieces and parts might be on the outside of it, I would use a scanning electron microscope. Now, the other one, our transmission electron microscope, is going to shoot electrons through the object. This is going to allow us to get a picture of the internal structures. Now, if we go back and look at the names that we've given these, scanning and transmission, if one means going through and one means just looking at the outside, I feel like it's a fairly logical thing to think that the scanning one is the one that looks at the outside, and to transmit electrons would be the one that goes through, which is the actual case. Part of the reason electron microscopes were not used for question 4 for working with living things is because if you're blasting electrons at something, you're more than likely going to kill it. You could put your hand under a light microscope and it wouldn't be a big deal. You could still see things. However, if you put your hand under an electron microscope, you're going to be missing parts of your hand pretty quickly. All right, for question 5, let's take a look at what we got here. Which microscope would you recommend a viral researcher use to identify the parts of a virus that can be targeted in making a vaccine? Well, let's see if we can get going here. First thing to know about this, the parts that help identify viruses and bacteria are called antigens. We talked about this in immune system, and these are located on the outsides of these cells and viruses. So based off of what we had said earlier about our microscopes, this one is more than likely going to be our scanning electron microscope. And again, this is related to the fact that scanning electron microscopes look at the outside of things, which is going to make it more useful in that type of a situation. Now, viruses are much smaller than bacteria, which means that the electron microscope would be very useful. If we were looking at something like a bacteria, which is a little bit bigger, and we still wanted to see the outside, we might be able to get away with a compound light microscope, but more than likely, our SEM, scanning electron microscope, would be the better choice. 6, which microscope or scopes would you recommend a coastal researcher use in trying to determine the best type of soil composition for a specific coral reef in the area? This one's going to be more than one microscope. If we're looking at soil and we're looking at the mixture of this soil, just looking at the top or the outside of it probably wouldn't be quite as useful. This is one where I would definitely be bringing in our light microscope. because as we analyze the different parts and split things up and just look at the general structures of what's there, we'd be able to get some use out of that. And on this one, we'd also bring in our transmission electron microscope. That's going to help us scan down. Actually, I'm not going to use the word scan. Uh, Analyze down through the layers of the soil. We're not just looking at the top, we're looking at the entire mixture of it. So making sure that we know what's at the different levels would be important and useful here, as well as analyzing some of the specific components using our light microscope. So those two would probably be our best choice for number 6. Clearing our screen. We move down to cell types. Now, I was actually really surprised that this is as small as this section is. Cell type always makes it to the EOC. Do we have plant and animal in a different spot here? I'm just going to look ahead just to make sure, because if we don't, I'm going to talk about that here also. I don't see plant versus animal, so we're going to do a little bit of an extra add-on part for this after we talk about, they ask it in 8, but I don't know why they didn't set it up like 7. So let's see what we got here. Similarities and differences between prokaryotic and eukaryotic cells. I'm just going to kind of make a list after these for things that they have in common or not in common, and we'll see how this plays out. Similarities between prokaryotic and eukaryotic cells. Cell walls. And in parentheses, I'm going to put some, because animals don't have cell walls, but plants do, fungi do, bacteria do, archaea do. Lots of cell walls and our types of cells here. Cell membranes in everybody. Cytoplasm. DNA. Ribosomes. Ribosomes are the one type of organelle that prokaryotic cells have. Just in case you forgot, the difference between our cells, prokaryotic cells are these guys with their DNA in there and a couple of ribosomes kicked around through here. These are our prokaryotic cells. These things look like a pill. Prokaryote, pill, we can kind of get that lined up there as far as the name for remembering which type these are. It's got a flagellum on the back, which is going to help it move. It may have cilia, which would be the little hair-like projections on the outside, also for movements. But these prokaryotic cells, they're very basic, very simple. They don't really have a lot going on, but they definitely need to be able to have ribosomes because they have to be able to make protein for themselves, and that is the job of these ribosomes. Now, as far as some differences between prokaryotic and eukaryotic cells, if we haven't said it yet, you are made of eukaryotic cells. Let's take a look at some of our differences here. Eukaryotic cells. EUK is euks. We're going to have things like membrane-bound organelles. And we'll give some examples here like Golgi, the ERs, lysosomes, vesicles and vacuoles. Really big one, nucleus. Both types of cells have DNA, but only eukaryotes have our fancy container to put our DNA in as our nucleus. There's a variety of other components that could be inside, but these are just some of the big ones that we've got here. With these differences, you should be able to get your points as far as the difference between prokaryotic and eukaryotic cells. Plant and animal cell, a little bit of a different situation. I'm going to try to draw some stuff here. So if we're talking about a plant cell, the first thing that we're going to notice is that our plant cells tend to be somewhat rectangle-ish. This is due to the cell wall that they have on the outside. It's going to have a nucleus, always does. It's a eukaryotic cell. But one of the things that plant cells have is they have this big lake that they keep inside here called a large central vacuole. That'll be one of the differences that we mention in a minute. I'm just getting it onto our screen here. For animal cells look a little bit more like a circle. Still going to have a nucleus on the inside because it's eukaryotic. So we've got our container for our DNA. And then we'd have our various other organelles on the inside. So we're going to make a little T-chart here for plant stuff and animal stuff. And see if we can get some of these differences. So in our plant cells, we're going to have things like chloroplasts. That's going to be helping as far as doing photosynthesis. Plants have cell walls. Plants have the large central vacuole. Those are some of the major differences as far as things that plants have. Animal cells, on the other hand, are going to have a couple things as well. The first one that I like to bring up because it's going to connect back to mitosis, which could make a huge difference because remember, any topic that fits more than one section is more likely to end up on the exam because it has more than one way of making it to the exam. Centrioles, sometimes called the centrosome. These are those little churro looking guys that split to either side of the cell and shoot out the spindle fibers to pull our chromosomes to either side in mitosis. Turns out plants don't have those. They do things in a slightly different way. Another thing for animal cells, lysosomes. Now, technically, I was digging around in our AP book. I found that most plant cells don't have lysosomes. They found lysosomes like in the root cells of a certain type of onion. But if we were to look at plant cells in general, we wouldn't find them there. So these are two major differences of things that animals have versus some things that plants have. Using those to help tell the difference will be really big. Notice that we gave chloroplasts to plants, and a lot of people go, oh, animal cells have mitochondria. So do plants. Both have mitochondria because both of them are doing cell respiration. That's a little foreshadowing for another question coming up later on. We're going to clear our screen here and head down to check out this vocab a little bit. Cell theory, our explanation of what cells are and where they came from. Light microscope, we talked about that at the top. You should be able to get that definition fairly easily. A stereo microscope makes it sound like it's something extra or special. It's not. A stereo microscope is a regular microscope with two eyepieces. You're looking through with both eyes. That's the stereo part of it. Prokaryotic cells, basic cells, no nucleus. And the only example of prokaryotes are bacteria. Eukaryotic cells, complex cells, yes, nucleus, plants, animals, fungi, protists, lots of examples there. And then an electron microscope, I think you've got this definition already too, but this is going to beam electrons onto the specimen instead of light. Hopefully this was useful to you guys. You'll be able to find the stuff that you need, and we strengthen some stuff up for your EOC. I will catch you in our next section. See y'all next time.